Good evening, everybody. My name is Tim Gibbs, Executive Director of the Delaware Academy of Medicine and the Delaware Public Health Association. I want to thank you for joining us, especially if you managed the migration from the old webinar room to the new one. We have a very large class size and actually exceeded our webinar limit. So technology happens. This is 2023 Delaware Mini Medical School. And I'm going to go ahead and run through some introductory slides. So about the program, Delaware Mini Medical School is a program of the Academy DPHA with co-sponsorship and support for this spring session from ChristianaCare. MiniMed has been running continuously since 2009. The 2023 session is offered as part of the Delaware Health Force Initiative of the Academy DPHA and is supported by state and local fiscal recovery funds through the Department of Treasury and the state of Delaware. There are no tests, there are no grades. There is also no continuing medical education or continuing education credit available. This is geared towards two audiences, primarily towards middle, junior high school, and undergraduate students, and secondarily, towards adult learners who are interested in learning more about how their body works, how we diagnose disease, and then how we treat it. Participants, and especially to you younger students who attend all 6 sessions live, will receive a certificate of achievement and participation, and that could be something useful as you move forward in your career. Program goals here are fairly simple. Number one, we want to enhance the health literacy in our community. The more we know about our bodies, how they work, the better we can take care of them. Second, as I said, we want to expose middle, junior, and high school and undergrad students to a range of topics and types of providers as a part of career goal setting. MiniMed instructors get to share their own career pathway with students to clarify how they got to where they are today as a practicing clinician or researcher. And also, this encourages a dialogue between students and instructors in a safe setting where you're not dealing with your own personal medical issues or you're about to get a shot or some sort of a diagnostic test. So this is a safe place. An action item, please. There are 2 ways you can do this. Right now, please either type your full name and email address that you used to get into this session tonight into the chat window. Or if you would prefer, you can email that information to nsabine, N-S-A-B-I-N-E @delamed.org. That helps us with attendance with a little bit of chaos that we experienced. This is the 2023 semester. We're going to start off with the very interesting and exciting developing field of gene editing. Then we'll do bariatric surgery, interventional cardiology, anxiety, neurointerventional surgery, and then general dentistry, and a much more complex oromaxillofacial surgery. This is sort of a little slide about who the officers and directors are of the Academy DPHA. Dr. John Swanson is our president. And at this time, I'm going to turn the mic over, so to speak, to Dr. Kate Smith, my colleagues, who will introduce this evening's speaker. Over to you, Kate.
Thanks, Tim. Perfect. So I do want to mention, it looks like chat is disabled, so I'm going to try and work on that. But for now, if you can email that email address that Tim was talking about to get your name in there, that's fine. Our speaker this evening is from ChristianaCare's Gene Editing Institute. Dr. Natalia Rivera Torres is a research scientist whose work has led to a fundamental understanding of how gene repair is carried out in human cells and how this process may vary in a variety of different patient samples. She is also credited with elucidating the mechanism of homology-directed repair in human stem cells in a process known as single-strand template repair or exact excision and corrective therapy. She is now the principal investigator of the Precision Genomics Group at the Gene Editing Institute. Her group focuses on identifying new clinically relevant genomic targets for which CRISPR-directed gene editing can be utilized as a therapeutic modality. And my hope is that she's going to give her talk tonight, and you're all going to be able to understand that paragraph when she's done. So, Dr. Rivera Torres, I'll turn it over to you.
Okay, thank you so much for that great introduction. Hi, I'm Natalia, and let me get my slides shared over here real quick so we can get this started. And yeah, definitely the goal for today is to get you guys introduced as best as I can do to the CRISPR revolution, especially in the healthcare setting. CRISPR is now being widely utilized from not only different sorts of background applications, but we're making really great breakthroughs into the clinic. And I really hope that you guys can get to understand that a little bit better, because it's not only about what career paths you can develop within this great innovative field, but also if you or any of your family members in any given time get asked the question, would you be willing to get this therapy, right type of gene editing therapy for yourself, that you guys can make a better educated decision on that, because those times are now, we are living them. So a little bit about me. I was born and raised in the small island of Puerto Rico, where I did my bachelor's in molecular and cellular biology at the University of Puerto Rico, Rio Piedras campus. And during my undergraduate coursework, I had a really interesting career path where I did research, but the research that I did wasn't actually wet lab work. I was doing research in being able to develop better educational modules to actually be implemented in the school system, but for nanotechnology. Why nanotechnology? I was actually bridging the gap that the Center for Advanced Nanomaterials had in the context of bringing my biology background into their other chemistry and physics heavy environment. And that way I was able to understand how can I learn science as a major, but find better ways to communicate science. Because as scientists, if we don't find proper avenues to relate what we're doing in our lab to the general public, then a lot of the science and progress is lost. And only what people can perceive from either TV shows, movies, and sometimes from general just news articles that covers a little bit in depth what we do, but it doesn't really give the context. So having better education formats for people like who are attending this platform today can get better exposition. I had the chance back to do my master's at Delaware State University, where I actually joined a lab for Dr. Eric Kmetch, which I currently still work with. There I started my adventure in gene editing. I started off with a platform that's not CRISPR related, but had a lot to do with the early works in gene editing. And then I was actually part of the discussions early on where CRISPR yet did not exist. And we were like, do we really want to start in this technology? Is this technology safe? Do we think this has a future back in 2012? And so I have lived the actual gene editing revolution of the discovery and implementation of CRISPR. And I was able to follow my career and do my PhD at University of Delaware, which I graduated in 2019 from part of the medical and molecular science program. But during my PhD education, I had the chance to work at ChristianaCare Health Graham Cancer Center, where our lab was located. So I started being exposed to the needs of patients. What do patients need? How can my science moving forward for my career have an impact in patients? I didn't want to do science that was just great for a publication, which we all want to achieve as scientists, but how can I make it to the bedside? How can I make what's already, how can I get to what's lacking for these patients better? How can we bridge the gap with this type of gene editing technology, specifically for cancer, which I'll do go in depth later on in my presentation. And then I spent a couple of years as a research scientist, PhD level in the Gene Editing Institute. And about 2 years ago, I was promoted to principal investigator, which now I have my own research team. And I've been working on identifying new targets early on where we can apply CRISPR to be utilized for a therapeutic modality. I did my PhD in pediatric cancer and trying to establish CRISPR models to better identify drugs that can be used for specific types of pediatric cancers. So I had already that niche growing within me, and working in the cancer center and part of the Gene Editing Institute, I was able to also start working on what we call the CCGI-001, which is a lung cancer therapy utilizing gene editing in the preclinical context. And I'll definitely close my talk with what does that mean. We are currently working for the pre-IND package. So we have been interacting with the FDA. So we have been making really great progress in that realm. So the gene editing revolution started back in 2012, where CRISPR made headlines in any type of context, not only in the science, but general types of news articles, National Geographic, Times, everywhere and anywhere you were hearing the word CRISPR. And it was I think it has been one of the greatest breakthroughs in gene editing for the last, I think, over 20 years at this point, where the fact is that the Nobel Prize of 2020 in chemistry was awarded to the two scientists that actually developed CRISPR genome editing to be implemented in human cells, and that was Jennifer Doudna and Emmanuel Charpentier. I am grateful to be working in a field such as gene editing, where it's predominantly women scientists leading the science within gene editing. It's not a norm. It's very rare to see women getting Nobel Prizes in science, but the greatest achievement in the last 20 years was actually awarded by women. So it's a really nice story also to follow on how we're making breakthroughs in science and promoting more women in STEM. So what does the Gene Editing Institute offer? We deliver scientific excellence in a patient care setting because we are part of ChristianaCare. And that represents the potential for us to really understand what diagnostics tools do we better need? How can we bridge the gap for precision medicine and therefore help and improve health? And our mission is for long-term healing to those who are hopeless or those who don't have good outcomes within the current treatments that are available. So we have two big aspects of research and development and also focus within the Gene Editing Institute. We have our translational science, which is very heavy driven for our CRISPR-directed gene editing for lung cancer, which I'll go in detail later on today. But also we have developed a program for education and outreach. And I'm not sure if any of you or most of you have heard at some point about CRISPR in a box, but we were able to develop an educational tool for high school students to be able to go through a gene editing experiment and therefore learn how to do gene editing as early on as high school. So when you guys are ready to go to college and undergrad and eventually, develop your careers in science or in medicine, you have already had that type of experience and understanding of, oh, this is CRISPR, this is how it works. I was able to do that. And we really want to make it something that we make it accessible for minority communities to gain access to scientific breakthroughs and pretty much just bridge the gap and make everyone available to that. So from the translational science, we've really have fulfilled our commitment. We are fulfilling our commitment. We've published papers. We make the science field grow as a whole by all the developments and all the findings that we have made throughout the different years and throughout the years and my career as well. We try to publish everything and anything that we produce in the lab as best as possible so the field can keep growing, because more people understanding and the more collaborative effect that we have within a field, the better contributions and the more the science grows. So as a lab from the translational science, we're doing really well, but we've also been able to fulfill our commitment of education. We've developed different pipelines to make the science better for everyone and accessible to anyone. from doing an in vitro system protocol that was one of our scientists' PhD program, to make that the commercially available kit that is CRISPR in a box, where we have now partnered with educational distributors such as Carolina Biologicals or VWR, and that kit is sold and commercially available for those teachers who want to implement this. And we've taken on a very aggressive mission to make it available for all high schools in Delaware. We also did an educational platform with a series of videos that we call Gene Editing 360, a video series, where we talk about the development of how gene editing platforms can be utilized in the case or specific study case of the series of sickle cell anemia, what challenges sickle cell anemia patients were facing and how gene editing technology could overcome those challenges, and why getting this type of technology and science understanding to everyone is very important. So like I mentioned, our education program has been really well established and is currently being developed, where we really want to make gene editing and CRISPR in a box and CRISPR technology available to all high school students and college students in Delaware. We have 3 core programs currently ongoing and working on. One is Train the Trainer workshop, where teachers from community colleges or colleges and even high school teachers can work with us, and we train them to better understand how to implement your CRISPR in a box experiments. We also have a learning lab, which we are currently finalizing, so we can have students come to Gene Editing Institute and learn how to do the kit itself at GEI. And we will also be working in the next couple of months to really establish a pipeline where if you as a high school have an interest of having one of us scientists or anyone from the Gene Editing Institute come and visit you and give talks and kind of pretty much go through the same process that we're going to do today, where we can talk about what we're doing in the lab or how we got to our career pathway, we can do that as well. So we are definitely open to interacting with you guys, making you guys more aware of opportunities that there are within the field, and really want to engage with the next generation. You guys are the next scientists. I am at a point in my career where now I'm building my team. So any of you who are here today could potentially be a staff scientist for me, or further on, you guys could even be a peer. So there are many applications for gene editing, not only from the context of how do we want to better people's lives and education and making things more available for everyone. So there are 4 main current research areas that I want to highlight today. That is drug development, animal models, agriculture, and gene drives. Throughout my talk, will predominantly focus on drug development because that's where my research is focused on and what the Gene Editing Institute is currently working on. But there has been great progress with the gene editing field to make these 4 categories really more amenable for all scientists. So we've been able to achieve through gene editing better animal models for human disease, to be able to better make universal transplant organs and better understand diseases such as Huntington's. Agriculture is a big field for gene editing. The whole conversation about GMO, non-GMO, when you have that conversation and you are looking into that, you're definitely talking about genetically modified organisms. So it goes into the gene editing context. How is this being applied to better make pesticide resistance for plants? Sustainable and storable food. We know that, world warming can, like the heat, everything is affecting all types of crops. So how can we be able to grow plants in different types of platforms that we aren't developing or have not developed yet? So that is where gene editing is helping. And also for gene drives, how can we help disease prevention? And the best example is malaria, which is propagated by mosquitoes, and we know that there's areas that are more prone to malaria. So how can we help better on our quality of life overall, and also from the context of control invasive species? So before doing a deep dive into gene editing, we have to understand the central dogma of biology. And this is, I'm going to start sounding like a high school science teacher, but this is really the core of understanding why gene editing and CRISPR technology is so efficient and has become the breakthrough science in 2020, winning the Nobel Prize. So we know that it all starts with the DNA, which is really what holds the key to pretty much all of us human beings. It's the basic way of our transcripts and our history and our nature being written. It's in those 4 nucleotides. And that DNA goes and gets transcribed into RNA, which later gets translated to protein. And what I want to point out is that most drugs that are available will interact with the protein level, and in some cases will interact at the RNA level. But where most of the problems lie for diseases, if not all problems, especially from the genetics context, it's at the DNA level, which is the basic unit. So if we only target the RNA and the protein, we're really not solving the problem. And that is what gene editing has been able to bridge the gap in, where now we can go to the root of the problem and treat it. So therefore, the persistence of the correction can be long-term, and we don't have to be utilizing so many drugs consistently because we haven't been able to go to the foundation of what is wrong. So there are 3 main divisions within gene editing. There's the cutting, there's the pasting, and there is the changing. And you can think about it from the context of when we're doing a Word document for all those write-ups and essays that you guys have to work on. It's kind of the same idea when we talk about the genes. When we want to cut, we refer to it as deleting, or in more specific terms, a knockout. When we're pasting, we're doing inserting, and we can create transgenic. And then when we're changing, it's also known as replacing or knock-in. And if you look at all these different contexts of the mice, when we do a deletion in the knockout, the mice on the right had its leptin gene, which controls for appetite. It was disabled, so the mouse does not know if when it feeds that it needs to stop because it has to find what his needs are. So the mouse just keeps eating and eating and eating because that gene is no longer there for control. We can insert genes into our genomes, and here there was a GFP, a green fluorescent protein gene, inserted into these pups to make, and this is the type of way that we do animal models, and these mice now glow green. And in the replacing or the changing, which is called a knock-in, by modifying just specific bases within the genome, we can now have the eyes of this mouse that had a GFP insertion, but in controlled manner, only expressing in his eyes. And these are the 3 main pillars of genetic editing. So in what I am going to focus throughout my talk today, and the work that I'm doing in our lab, we're specifically working with the deletion and knockout of a specific gene in the context of lung cancer. So why it is important to understand DNA, and how can we take advantage of the natural processes that happen in our DNA to make gene editing more efficient? So there's 2 main pathways which DNA constantly is going through. It's called the non-homologous end joining and homology-directed repair. And this is constantly happening because there are environmental factors, biological factors that produce breakage of our DNA. But when it is joined in a non-homologous way, we can produce gene knockout and mutations that disable genes. But when we can control this repair, and we can either use templates or band-aids that can lead the DNA to repair itself in specific ways, we can actually produce those modifications that I talked about from the insertion or just the basic knock-in changing. So how does this go in line with what gene editing is in the context of the particles? What is gene editing, and how are we going to relate to it today? So today I'm really going to focus my talk from the CRISPR-Cas9 system. And what this means is that there is a molecule that is composed of 2 units. A CRISPR-guided RNA, which is represented by this gold man here, and then the Cas9 nuclease, which is represented by the scissors. So what CRISPR-Cas9 does is that we can design a specific site within the DNA, within specific cells, where we can tell where do we want to cut the DNA. And previous to CRISPR-Cas9 implementation and discovery and development, we were able to cut the DNA. It was just in non-precise forms, which led to other side effects that were not intended. Therefore, the technology was never really taking off as much, and it was very hard to control for the specific outcomes that we were interested. But by having this gold man here, we can tell it at what specific base within our genome we want it to cut. Therefore, we were able to really break those challenges and be able to define and control to the most minimal aspect possible to make this the most efficient way of modifying DNA. So how is CRISPR and in nature, how was this, where did this come from? Did someone create it in the lab? Well, it was really interesting that back in 1980s, there was a scientist who had realized that bacterial cells actually have their own defense mechanism against viral infections. And he was able to identify what is now known as CRISPR, because originally, when a bacterial cell is infected by a virus, the viral DNA integrates into the host genome because it's actually breaking down and chopped off. So by doing this, the bacterial cell actually creates its own memory. So when it's re-exposed to these types of viral infections, it already had the mechanism of actually preventing an infection and preventing itself from getting sick. So these bacteria were actually the key to understanding the CRISPR nature. And what Donna and Charpentier did was actually repurpose this. And there have been many instances where biological systems have been repurposed to make it more amenable for specific use, such as it is here with CRISPR. So human genome editing, ethics, and policy. I don't know how much of following the CRISPR revolution or gene editing revolutions you have been aware, but there have been some negative connotations within the field of scientists just not really doing what they should be doing. And unfortunately, this has dampened the field for a couple of years, but now we're coming back out of those dark days and we're in brighter days where we have better defined ethics and policy in place to prevent people from going wild and crazy. So genome research is definitely it's going to change. It has revolutionized everything as we are working now in the biological era. So with the CRISPR, we can tell the body where to heal itself. Sometimes this involves correcting a genetic mutation, such as sickle cell disease, and there are actually ongoing clinical trials where they're utilizing CRISPR and gene editing technology to treat sickle cell patients. And there's other times where we can mark a gene for destruction because that gene is blocking a therapy that treats a disease, such as I will focus on later on today for lung cancer. So we have the power to tell the body where it heals itself. And with great power comes great responsibility. And I know that Hollywood has not done justice to what we do in the lab. And I don't know how many of you have watched any of these movies or have heard of any of these storylines, but what they present here as gene editing and utilizing CRISPR, and it is to a certain point true, yes, we have capabilities of modifying the DNA, but we can't truly make, gorillas that are going to be, King Kong size, and we can't really make dinosaurs exist again, or less alone, start producing, Spider-Man in every corner of the world by modifying spiders. So how can we regulate genome editing so scientists and people don't try to do all these things that Hollywood is trying to present that we are able to do? So this conversation is not a new conversation. This conversation actually started back in 1982 where we understood that there was the development of gene editing and there was great potential for this type of technology to be utilized in the healthcare system. So there was a President's Commission for Ethical Study and Medicine to really define the parameters of how will we control in the future and how will we emphasize policy to make sure that scientists properly develop platforms for healthcare in a safe manner, and also in a manner where everyone can get access. So there's governance principles for genome editing, and this was decided by the National Academy of Science and the National Academy of Medicine came up with these principles, promoting well-being, transparency, due care, responsible science, respect for persons, fairness, and translational cooperation. These are the principles which all scientists that want to take their science into the clinic need to be aware of and need to take these into consideration. We want to have technology that is developed for everyone and anyone, not for only certain types of backgrounds or certain types of diseases. We want to be able to have it fair and transparent where we understand what the technology does so people can consent to do if they want to have these type of treatment or not. But most importantly, that we promote well-being, that it's not just for the sake of growing the science that we develop therapies to say, oh, well, we weren't sure what it was going to do, but we thought that it was a great idea. We're taking really well thought processes to developing our gene editing lung cancer platform at the Gene Editing Institute to ensure that we're promoting the best well-being of any type of lung cancer patient that potentially could be treated with our therapy. And that goes hand in hand with responsible science. And if anyone wants to go into the scientific field, you definitely want to be holding true this principle of responsible science. So there are two ways as we can do gene editing. There is a germline, and then there is the somatic gene editing. Somatic gene editing is any type of edit that happens in any cell in your body that are not related to a sperm or an ovule. So what this means is that when these cells are actually edited, we cannot pass it down to our progeny, meaning that the risks of any unnecessary or uncontrolled mutations cannot be passed into next generation. And that is where the consensus has lied in which types of cells are we truly working to do gene editing at this time. We're not doing a lot of work, right? If I'm understanding, there isn't any work at all right now doing germline editing because we don't have a well understood context of what does editing these cells do long term? What happens if these mutations are passed to our progeny? We won't be able to control for any unintended effects, and therefore there has been moratorium and there has been policies put in place where scientists cannot be testing these types of drug developments. But that does not hold back the field. We are still working a lot, and we've actually looked at how do people feel? What would people say if they were having a baby, right? And we could utilize gene editing to treat these types of congenital diseases. What would the general public say? So treating a serious disease condition that a baby would have at birth, 72% of the general public think that it's appropriate to use the medical technology. If we're to reduce the risk of a serious disease that could occur over their lifetime, 60% of the people agree. And from the concepts of what if we could make someone more intelligent, right? 80% of the population or of the tested inquired population here said that they do not agree. So this is really where we stand. We want to make people's lives better, but we don't want to go down the road where it's almost to a point slightly unethical to be able to decide if you want to make someone more intelligent or not. Let me see if I can change the slide. Okay, so gene editing in the clinic. So we have two lines of research ongoing for the gene editing. There is an ex vivo, and if any of you have heard about CAR T cells for leukemia, this is where it falls in the category of ex vivo, where cells from the patient are actually collected. In the case of leukemia, blood cells are collected. They are treated with CRISPR or gene editing technology to make or change the specific genes to express certain types of receptors or stop producing a certain type of protein. And then those edited cells, which are filtered, go injected back into the patient. And therefore now the patient has a population of edited cells that can grow and therefore make the patient better. And there's a sense of in vivo editing, which is where we in the Gene Editing Institute are developing our platform for our lung cancer work, where we would deliver the gene editing molecules, the CRISPR-Cas9, into the patient either by an IV or a direct injection into a target organ, or in this case, for our work, into a specific tumor, where the editing happens within the body. So these two realms actually have ongoing clinical trials. These are just the highlight that I wanted to present today, but there are many more clinical trials ongoing where they're utilizing some form of gene editing or a CRISPR-Cas9 variation for different types of disease backgrounds. If you look at the last one right here, it says the knockout of the TTR gene in hepatocytes with patients using a CRISPR-Cas technology. This is the actual clinical trial where they're actually delivering through a IV setting, and I'll go into details a little bit about why I want to highlight this specific clinical trial. But it is one of the most recent clinical trials where they are doing in vivo editing, not so much ex vivo. So CRISPR in the clinic, the lung cancer case study, and this is the work that we're doing at the Gene Editing Institute. So lung cancer is where your cells in the lung mutate, growing uncontrollably and clustering together to form a tumor. And there are many causes of lung cancer, either from smoking, radon exposure, other hazardous chemicals, particle pollution, and finally anything that could be your genes, hereditary. If your family has a history of lung cancer, you could also be potentially a lung cancer. You're at risk of developing lung cancer. So why did we want to go after lung cancer? We're part of ChristianaCare, and back in, I think if I'm not mistaken, 2017, when we were already part of the ChristianaCare Health Graham Cancer Center, we wanted to find a project that from the Gene Editing Institute, we were working side by clinicians. We wanted to make a project that was relevant. We wanted to hear from the clinicians what they thought was needed, not from us as scientists as, oh, we found a gene, and then going to the clinician saying, do you think this is manageable? Do you think this would be something that you guys would want to know a little bit more? We wanted it the other way around. We wanted to know what was lacking in a specific type of cancer that we could utilize our technology to bridge the gap in therapy. And our clinicians at ChristianaCare, specifically the director for the cancer center, Dr. Petrelli, said, I think you guys should look at lung cancer. And lung cancer is the #1 cancer killer in worldwide at 29%, and in the US as well. It's the leading cause of cancer mortality here in Delaware, with 540 deaths reported in 2019. And it's the 3rd highest cancer incident rate in Delaware, with 840 new cases just in 2019 alone. And I want to really highlight the incidence. It is the 3rd highest cancer incident, but unfortunately, most, if not all, lung cancer patients that are diagnosed are either at a stage 3 and stage 4. And what does that mean is that it's a very late stage cancer. So the diagnosis and prognosis for these patients to outlive their disease is very, very low. So normally they present when they have a systemic systemic development of their cancer, in most cases already in metastasized, where it's very hard to control. So lung cancer symptoms usually not appear until the cancer has spread, just as I mentioned, to other parts of the body. And unfortunately, it's very hard to treat. But ChristianaCare has a platform, and it has a campaign where it's offering CT scans to do lung screenings, and these have proven to detect cancer early. So not only is ChristianaCare taking on the mission to better diagnose patients at early stages, which we can better treat, but also for those late stages, from the research concept, to develop a technology and a therapy that we can utilize on these late stage cancers, specifically lung cancer. And it was very important also to work in lung cancer because there has not been a lot of progress. It's been more of a slow, steady progress in the type of treatment that there is available. Back in the 1970s, the development of surgery to be able to really take out all these tumors. But if you are getting most of your population diagnosed with metastatic cancer, you can't really operate. So surgery is not a candidate for any patient that is in late stages. It mostly will be utilized for early stages. So if most of our population with lung cancer is a late stage, we needed to develop treatments that are more amenable to that population. Radiation was an option, chemotherapy combinations, but we know now that chemotherapy is very harsh on the body. It works efficiently in killing cells, but it doesn't only kill the cancer cells, it kills any type of cell which is exposed to these types of drugs. So in the early 2000s, targeted therapy started being developed because the researchers and the clinicians knew that we need to find better ways to treat specifically the cancer cells. And there has been slow progress, but unfortunately, there still has not been a way to really define until CRISPR and gene editing came available to be able to discriminate between cancer cells and non-cancerous cells. And that is really the focus of where our gene editing platform is developing. We have been able to identify a therapy where we can only and exclusively treat the cancer cells, and therefore we don't have to have those negative side effects that some of these cancer treatments have, specifically chemotherapy, which is the standard of care for most late stage cancers. So there's different categories of classification within lung cancer. There's the small cell lung carcinoma and the non-small cell lung cancer carcinoma. The platform that we are developing is specifically for the non-small cell lung carcinoma. There are other labs and other technology being developed for the small cell lung carcinoma, but in this context of today's presentation, I will be focusing exclusively on this non-small cell. Our early work was developed doing in adenocarcinoma, but we were able to broaden our spectrum and really define a better niche for our work. And we ended up currently focusing on squamous cell carcinoma. And in the later slides, I will explain specifically why squamous cell versus adenocarcinoma. So we knew the disease that we could go after, which in this case was lung cancer, but we needed a better definition of what we could do within lung cancer. And we were able to find within the literature that there was this gene called NRF2, and that gene is actually responsible for chemoresistance in non-small cell lung carcinoma. What this gene does is that when you are exposed to different types of oxidative stress, this gene actually starts being produced or produces other backgrounds and activates other genes in other pathways that are supposed to help you detox your body from all these chemical harshness and all these really bad things for your cells. And what had been correlated was that in certain types of lung cancer, the pathway that regulates this gene was actually mutated, and therefore the lack of proper regulation of the gene leads to chemoresistance. So the idea was that patients that have high NRF2 were in late stages, meaning that NRF2 expression is associated with a worse overall survival, and not only survival, but least importantly, for the context of how we wanted to manage the disease, it was that there was less reoccurrence when this gene was properly maintained. So when this gene is not maintained properly, there's higher reoccurrence. So more patients were not being able to survive not only their treatment because they had to go through higher chemo doses, but also the type of cancer was much more aggressive, therefore leading to more metastasis. So what the graph here is saying is that they measured the mRNA, which is the way that in some cases we are able to measure if a gene is being highly expressed or not. And they did all these 47 patients, so 1 through 47, each one of them is a patient, and then they were compiled by different categories. So what they define as normal were patients that were not presenting any type of cancer background. But then they had a stage 1, stage 2, and stage 3 patients, and they measured how is this NRF2 being expressed in these patients, and they found the correlation that the later stages had this association of this gene being highly expressed, not only being highly expressed, but they were chemo resistant. So we went and decided to go to the drawing board and say, okay, we have identified a gene. How can then we utilize CRISPR or gene editing as a whole to be able to, do something to this gene so it's no longer making these patients chemo resistant? So the idea is to utilize CRISPR to disrupt the gene and turn it completely off, where now the cells are more amenable to being able to tolerate chemotherapy and therefore die, and you can produce a tumor shrinkage. So on paper, most of the time the science sounds great, but it's in the practice. Were we able to really prove what our hypothesis was? And we in fact were able to. Back in 2019, we published our first paper on NRF2 and how this is the start of what we are now working on for cancer therapy. We were able to show that when we had cells that had gone through CRISPR gene editing, and NRF2 was knocked out, or this gene was no longer dysregulated, the tumor shrunk and the cells became more sensitive to chemotherapy. And how did we do that? Most of the time in science, the way that we prove these types of biological assumptions from the hypothesis testing when it comes to clinically relevant clinically relevant approaches is through animal models. So we grew cells, cancer cells that went through gene editing and cells that did not go through gene editing. We injected them into mice and let these cells create a lung cell tumor. Once these mice had the tumor, we started chemotherapy treatment as our patients in the clinic would do. So at day 0, we had chemotherapy. We also had another round of chemotherapy at day 3 and another round chemotherapy day 6 and the final chemotherapy round at day 9. Because we know that in the clinic, patients go through multiple cycles of chemotherapy. It's not just a one-time exposure. And what we were able to see and prove was that if you look at this panel E down here, is that when we knock out the gene NRF2 and expose these edited cells to chemotherapy, the tumor in fact shrinks. So the top that says A549 is our lung cancer cell line that did not get exposed to any gene editing. And cisplatin is one of the chemotherapy reagents that lung cancer patients get exposed to. These tumors are very big. But when we have edited cells that have been exposed to gene editing where the NRF2 gene is no longer active, these tumors actually shrank in the presence of chemotherapy. So we were able to prove that we could restore chemosensitivity with NRF2 knockout in tumors. But at that time, our gene editing platform could not really discriminate we could target very efficiently at a specific site within this gene, but we could not make the discrepancy between cancer cells and non-cancerous cells. And that just fed back into the loop of, well, chemotherapy can also reduce tumor growth in certain contexts and in certain cases. Okay, we know now that we can overcome the cases that have chemo resistance, but how can we better the quality of life of these patients by not affecting all of their cells? Because from a context of gene editing, we do not want to be editing all the cells. We only want to edit specific types of cells, in this case, only the lung cancer cells. And one of our, at that time, graduate students who has now become another principal investigator at the Gene Editing Institute did a literature search, and she found that there are specific mutations in the NRF2 gene in the context of cancer backgrounds that produce specific CRISPR recognition sites. So not only can we better treat a specific defined population within lung cancer or different types of cancers, but we can find the discrimination for utilizing a CRISPR technology or a gene editing technology that could only affect the mutated cells and not the normal cells. So we found that there is a lung squamous background which is considered 14% of this population, that has a specific mutation that we call R34G, and that is in the context of just genetic notation, where this change of a amino acid produces a new recognition site for CRISPR technology that is not present in the non-cancerous cells. And not only is this mutation associated with the lung squamous background, but it's also a high predominance percentage in esophageal, uterine, endometrial, bladder, urethral, head and neck, and cervical cancers. So if we could develop this technology to hold true within the lung cancer background, which is really what we were focusing our efforts and are focusing our efforts at the time, and show this holds true, we could expand it to different types of cancer backgrounds. So we could be developing a technology that doesn't only apply to one cancer, but multiple cancers, and also a technology that if we can identify these specific CRISPR recognition sites in other genes, we can develop better platforms that control the gene editing and that can discriminate between our intended cells of treatment and our non-intended cells for treatment. So when I talk about this CRISPR-specific gene editing, this is the representation. So the top is what we call the mutant NRF2, which is the R34G. So in order for our CRISPRs to cut, it usually has to have a recognition site, which is called a PAM site, and it's a protospacer adjacent motif. And what these recognition sites, it sells this Cas9 protein, which are the scissors, as I mentioned earlier in my talk, that say, oh, this site is the specific site that you can cut. Go ahead and do so. So when these sites are present, the CRISPR can actually produce an intended cut in the DNA. When they're not present, as it would be in a wild-type NRF2 or a non-cancerous background, the CRISPR will not have that site, so it cannot cleave. And our research scientist was able to show that, in fact, this holds true. So why did we do the switch more specifically into the lung squamous cell carcinoma and not find a better defined way to apply this technology to the adenocarcinoma background? And it was because for lung squamous cell, there is less studies, so it's a background that needs more development. There's not a lot of treatment options. The therapy that's available, unfortunately, has failed for targeted therapy, so specific type of therapy are not available. And these patients have very poor response and aggressive prognosis, so it means that this type of specific cancer is highly aggressive. And the overall survival rate of these patients is less than 15 months. So we really wanted to work within a niche of lung cancer that had a need. we know there are many drugs that work well out there or can expand the life of people who have these types of cancers. So we wanted to give the opportunity of the patient population that doesn't have these available drugs that have been developed a chance to also have an extension of life. We're not trying to cure cancer because that there's still much more to understand and really learn about cancer before we can use the word cure, but we want to be able to minimize the load or the disease progression to a point where a patient can now, or tumor burden where a patient now can go into surgery, or they can live life milestones where they can go to a graduation, they can attend a wedding, they can have those special moments that they can achieve and outlive. Keytruda, which is one of the lung cancer drug that has been on the market for a couple of years now, and I'm sure at some point most of us have seen the commercials on TV and everything. Keytruda only expands a lung cancer patient's life by 3 months. So if we could develop a technology that could expand the lifetime of a patient for 3 months, then we are fulfilling what is already in the market, and that is something that is not available yet for lung squamous backgrounds. So here is our principal investigator, Kelly Banas, along our direct gene editing direct gene institute director, Dr. Eric Kamech. Kelly was the student who found that these specific mutations in different in the NRF2 genes create CRISPR recognition sites, and she's been able to prove through her work and has been the base of all of our gene editing FDA technology that we're currently therapy that are working on, that editing this specific site will actually make the cells better and more vulnerable to chemotherapy. And we've published a lot in this background where we've taken the time to understand the mechanism of gene editing and how we want to take this approach into patients, and not only done it from the mechanistic standpoint, but also from the relevance of the clinic, because we really want this technology to succeed. A lot of times, breakthrough technologies are not well studied and defined, of understanding the limitations or advances that we could do from the early science before it's taken or promoted to go into a clinical setting. And since we want to move this as quickly as we can and as efficiently as we can, we have dedicated the early years of this work to really define the mechanisms, and not only define the mechanisms of CRISPR gene editing from the context of technology, but we better understand the role of NRF2 and what does mutating or knocking out this gene truly do, and will it hold true when we move into the clinic. So that's going backwards, and there we go. Okay. So there are many challenges that need to be overcome in order to be able to take gene editing into the clinic. And one of the major limitations and challenges that the field has faced is, how do we efficiently deliver our gene editing technologies? We can deliver gene editing very efficiently into cells and in vitro defined context, but unfortunately, we've been facing many barriers when it comes to the in vivo setting to scale it up, but be able to define where specifically we want to deliver this. So routes of administration have been a way to really work on that delivery. Is it being distributed to all tissues? We don't want it to go everywhere. We want our technology to be defined in a concise space, because if we can have the discrepancy of being able to identify mutated or non-mutated, or where it makes the edit, we don't want it to go everywhere. There are studies that have shown that if for certain types of delivery, specifically from the viral vector delivery standpoint, it could produce an immunological response. If we want to do a type of treatment that is going through IV or systemic, and we want it to be like a brain type of treatment for brain cancer or any type of other neurological context, we need it to be able to pass through the blood-brain barrier. So how can we make the delivery efficient to pass through blood wall vessels? And then just the cell as a whole, the cell is not going to want to be able to take in anything that it senses that it's going to potentially harm it or make it die. And when it comes to the way that we're applying our gene editing technology, where we want to mutate the DNA in the cell so the cells eventually start dying and then that tumor starts shrinking. But we've been able to pretty much overcome most of the challenges for our gene editing therapy, and I'll go a little bit into detail of how. So when we talk about genetic medicine challenges, just like I mentioned, delivery is really what is going to make or break if you are going to have an efficacious drug. And a lot of work early on, and in some cases, some clinical trials and a lot of science is being utilized by or developing viral vector delivery. But these viral vectors come with major limitations. They are high immune response. They can potentially produce unintended genomic insertions. They limit the size of the gene or the payload that you want to deliver. And our CRISPR-Cas9 technology is very big. So we would have to use multiple types of viruses or multiple types of carriers in order to just deliver one molecule. And then there's the cell culture required. So scaling up of capabilities are just too heavy and would take a lot in order to be able to produce these types of therapies. But lately, there has been a shift in the field to walk away from these viral vectors, which were the early on days, to now shifting gears to what is known as a lipid nanoparticle. And the advantage of using these types of particles is that they have low immune response. There is no potential genomic insertion or unintended edits to your DNA by just the vector itself. We can pretty much carry anything. There is not much of a size limitation, and it has very low potential of immunity, and they're produced synthetically in the lab. So doing scale-ups and thinking about manufacturing is not an issue. And in fact, the COVID vaccines, the way that they were developed for delivery is utilizing these lipid nanoparticles. So they have become highly relevant to our lifestyles and life day-to-day. So back in 2021, 2020, this article came out in the New England Journal of Medicine. And when you work in translational research, this is definitely one of the journals to keep your eye out, because most of the cutting edge technology, most of the cutting edge therapy developments will be published in the New England Journal of Medicine. So if any of you want to have any heavy science-driven reading, definitely look up the New England Journal of Medicine. They really have everything and anything that you would like to see that is ongoing currently. And then all of the basic science for clinical trials is published here. So in 2021, they published a paper on CRISPR-Cas9 in vivo gene editing for transthyretin amyloidosis. And the reason why I want to highlight on this paper was because this was one of the first clinical trial developments where they were utilizing these lipid nanoparticles to deliver the CRISPR-Cas9 system in the most efficacious way that we have seen yet. They were doing a systemic delivery, so that means that they inject through IV a suspension that has these particles that contain the guide RNA, which is specific for the TTR gene, and then the Cas9. And what they were trying to do was actually mutate or knock out a gene for the liver, for better liver, for better liver output. And it really proved the point that utilizing these lipid nanoparticles was the most efficacious way to do so yet to date. It definitely showed that there wasn't all these immune response. We didn't have to worry about timing or dosing in the way that we had to scale up with the viral particles. This has just opened the door for us to really move forward. And so far, it's holding true that the safety parameters that these early on clinical trials are testing have been able to be overcome. So it's showing that another level of safe use of CRISPR and Cas9 technology in the context that we are trying to utilize it in the Gene Editing Institute, where we want to knock out a gene to better people's life. So in our case, we will not be utilizing an IV administration or delivery of these LNPs. We will be utilizing an intratumoral injection through bronchoscopy, where the patient is slightly sedated, and through this bronchoscope, we can go specifically to the tumor and through a site-directed injection, deliver the LNPs, and therefore we can target specifically not only the tumor, but as I mentioned before, when we talk about tumor microenvironment, there are the normal cells that still compromise the organ tissue from the lung, but we really only want to target the cancer. So by these LNPs, our specific site within our tumor, and with our CRISPR-Cas9 that will only affect our tumor cells, we're therefore not having to worry about unintended edits or our CRISPR-Cas9 spreading through other tissues that we don't have interest in targeting. And it's a very controlled and defined aspect, and it's a very well-defined controlled therapy as well. And this is the approach that we plan on doing when we get into the clinical setting. So we're not only needing to not worry about delivery development, because this is a pretty self-established system where these types of intratumoral injections and bronchoscopy is really well developed and established in the clinical setting. So far from our clinical pipeline development, we've been able to show that not only is our CRISPR gene editing working very, very efficiently, but also that we're not spreading it to other tumor or other organs within our samples. So the in vitro context, what this means is that with 3 different LNPs that we're currently testing, in this context, we're going to refer to them as LNP 3, 4, and 5. The percentage here on the left means how well is the gene editing, or how well is our CRISPR technology working. So the higher the number, the better. And in this case, we're achieving over 90% of editing with LNP delivery. In the past, we had to use different types of delivery mechanisms, and our maximum point of really editing this NRF2 site was only being able to achieve efficacy about 50% or 70% on a good day. But now we can robustly and routinely overcome that and get all the way to 100%. So we know that this LNP can definitely deliver the CRISPR-Cas9 into cells, and we can edit the gene. We can knock out NRF2. But when we were looking at the context of in vivo, is this intratumoral injection, and are these LNPs in fact only going to stay within our tumor, or do we going to have to worry about any spread of this LNP or of our technology going anywhere else? And when we did the in vivo modeling, where we injected intratumorally our LNPs, we were able to only detect the LNPs that carry the CRISPR-Cas9 in the tumor itself. We looked at the lung, just normal tissue, the spleen, brain, ovary, and liver, and there was no LNP detected. Therefore, we're assuming, or we have looked a little bit more deeper into the data, but for the context of this presentation, I'm only just focusing on this type of data where there is no spread of the LNP. We have looked at, is there Cas9? Is there other of the CRISPR technology there, and we have not been able to pick it up. Therefore, our technology can discriminate between the normal and cancer backgrounds. We can define it to a specific site, and we are using a delivery mechanism that is already in the clinic and has shown to be safe and efficacious for treatment in patients. So there's still a lot of work to be done. We're currently working on doing proof of concept experiments where we will be taking our CRISPR technology to edit tumors in mice and then expose these mice to chemotherapy to be able to see if we can capture the tumor reduction, just as we have done in previous studies, as I showed earlier today. And then once we have all that data, we will be going back to the FDA for our pre-IND meeting, and then we can go into IND, and hopefully within the next 4 years or less, we could be potentially treating patients in the clinic. And this here is the Gene Editing Institute staff. This person right in the middle is Dr. Eric Kamech, our director. The Kelly Banas is with the pink shirt, who is the scientist and principal investigator who identified this specific mutation that we're now developing to be able to discriminate cancer and non-cancer cells. And the group as a whole, we're located at the STAR campus, where we have our lab there. We originally were in the 4th floor of the Cancer Center, but we moved out 2 years ago, and now we have the Gene Edit Institute independently established. So if any of you are near there, make sure to come and visit us. And with that, I will open it to any questions. I hope that you've been able to learn about gene editing and CRISPR technology and how we plan to utilize it in the clinic for lung cancer and any other type of cancer background that applies to our specific mutation. I can take any questions if there are.
All right, there are a couple of questions in the Q&A.
Okay.
And I think I'm going to start at the top. Is gene editing used to heal people?
So when we talk about healing, we talk about modifying genes that can make a person, to try to help a person's quality of life. I can say that when we say about healing, it's from the regular concept of healing. Now, when we think about the context, for example, of sickle cell anemia, right? Sickle cell anemia patients go through crisis, which means that their blood starts accumulating because of the sickling, and that produces a lot of pain. With this type of treatment that is ongoing with CRISPR and gene editing technology, we can increase back up the expression of fetal globin. So therefore, the cells should no longer be sickling or sickling as much. So the level of crisis or the amount of crisis events that these patients go through are less. So their quality of life is better. And I'm sure that for them, that is a type of healing, but it's not completely eradicating the disease. So we're trying to heal, but it's more of management of diseases.
Thank you. Next question. Can gene editing be used to eradicate learning disabilities or mental health issues passed down by DNA? And would it be an ethical debate on that?
Yeah, I think when it comes to behavioral change, anything that would fall in the context of behavioral science, neuroscience, I think that would definitely be an ethical debate. Because we do know, and there is still a lot of research ongoing of understanding how these genetic mutations or methylation patterns affect our behavior, but I don't think it has been fully established where we can say that these defined genetic mutations will make a person's behavior be specifically in this type of context. There are a lot of environmental aspects that come into play, and then it goes into the ethical conversation about then when do we screen for these types of mutations? Would we screen babies at the time of them being born to better know what predispositions you have? Right now, genetic testing is used for very defined and specific contexts for more like cancer, hereditary cancer, family known hereditary cancers, or genetic mutations or genetic diseases as a whole, but not from an informational context for behavior. And unfortunately, genetic testing is very expensive, and we still have to have better defined conversations that if we were to use it for behavior, how would we manage costs and how would we control the data that is available? Would you want everyone and anyone to know that you've been sequenced and that you have these specific mutations that could lead because from the behavioral science context, it could lead to that. We haven't been able to prove that it's 100% going to affect or you're going to have that behavior outcome. I hope I answered your question.
I mean, it sounded good to me, so yeah. Question in the Q&A, what kind of classes should a high schooler take if they want to go into this line of work?
So from at the high school level, I definitely want to say this is a very interdisciplinary field. You want to be exposed to all your basic sciences. So do not, run away from taking your regulated science courses. But if for the gene editing, you definitely want to have a very solid genetic background. So the more exposition you get to genetic context or genetic classes as a whole, definitely do that. But do not limit yourself to that. We at the Institute have scientists from all different backgrounds. We have one of our principal investigators, actually her undergraduate was in chemistry. One of the other principal investigators actually has a criminal science background. That's where he started off, and then he did a master's in molecular biology, and that's why he now can work with us at the principal investigator field. But having all these different types of exposures and people with areas of expertise or areas that you like makes your science and makes you as a scientist to just approach things differently than the traditional streamlined, mainline, narrow viewed biologist. And I think that's what really makes the field better, is that we're very just taking ideas from any context of where could we find other CRISPR molecules. So maybe a microbiologist could help us do that. So I think anything that is relevant to biology for sure. And the more molecular, the better, because this is such a molecular heavy field that you want to be able to really understand, but it is not limited to that. So I would say just broaden your aspects and of anything and anywhere, that class that leads you to any exposure that can come back to molecular biologics. Yeah, that's definitely molecular biologics.
So there are a couple of questions in the chat, so I'm going to try and make them into one question.
Sure.
So in general, one question would be, besides lung cancer, what other types of cancer can gene editing work on? But there have been questions about specific diseases. Can you treat juvenile hyaline fibromatosis? So could you maybe talk about the types of diseases that gene editing can help and what they kind of have to be like in order to get that help?
Sure. So with gene editing, we can pretty much edit any gene, right? Or any site within a gene or any site within our genome that we would like, as long as we have that specific recognition site. And there's multiple of them throughout our DNA, and there's different variations that the different types of gene editing molecules require, so you can play around with that. But it depends on the type of editing that you want to do. So for example, in sickle cell anemia, there has been two approaches done. Sickle cell anemia is produced by a specific base change. So almost like the mutation in the lung cancer where it produces the change in amino acid. The same thing happens in sickle cell anemia. But in sickle cell anemia, we don't have a specific, it doesn't produce a specific recognition site for CRISPR, but we know the site needs to be edited. So we just cut it as a whole. So the same can apply for any gene, any disease. It's more of what you need to regulate from the disease context. Do you need, is the protein affected? Is it a faulty protein or a gene that produces faulty outcomes? Is it a mutation where part of the gene is completely missing because you have a rearrangement? Or is it the context where you want to make more, for some reason your body's not producing enough of that specific protein, or you need more copies of that gene to be expressed. So there are different avenues of utilizing gene editing for different disease backgrounds. I think the most focus right now is in developing technology or developing therapies for genes that by eliminating that specific gene that is mutated or dysregulated, there is a clinical positive outcome where we can make the person's quality of life better when we eliminate that faulty gene or that gene as a whole. But there is no, I think, limitations yet that I'm aware where I can say, oh, for this type of disease from the genetic DNA context, we could not utilize CRISPR. I think that is why gene editing is dominating the biology field for model development, is because we can produce variants of these different types of diseases that we could not do before. So we can utilize gene editing as a therapy, but we can also utilize gene editing to better define the context and involvement of different genes in disease backgrounds. We might know that a specific type of gene is affected in diabetes, but because there wasn't a technology where we could define and specifically edit in a very limited way, we couldn't know what happens if we target that gene, meaning we disable it or we make it be expressed more. So now with the gene editing technology, we can create better model systems, and then we can also develop better therapy, more disease-specific therapy, not broad spectrum therapies. That is one reason why a lot of drugs that are available for different types of genetic backgrounds, they're good at mediating certain types of symptoms, but again, not going down to the root cause, because these drugs might not be going against the faulty gene or the site that really needs to be targeted. They're more developed towards mitigating and managing specific symptoms that can be sufficient for a better quality of life. Therefore, they become a standard of care.
Gene editing be done in utero, or is it more of a ethical question at this point?
I think that's definitely going to be an ethical question. Anything that is going to be in utero is a very fine line of what is accepted in the within the scientific context and whatnot, because there are a lot of limitations of us studying genetic modifications and even exposure to drugs as a whole at that level of human development, where we have to really prove that there is a benefit in the quality of life to a very, very, very far extreme that there is no other option. But unfortunately for DNA modifications, we're not there yet. That's why a lot of there is a lot of just precautions of women who are pregnant, right? They do not try to not take X, Y, and Z medication, and it's not because it can harm the the baby or the fetus, it's because there hasn't been studies done to know if that exposure will harm the fetus or not, because there are very defined parameters that signs can be taken specifically from the in utero context of it.
So there's a question that I like. What does an average day of research look like for you?
Yeah, so that's a fun question. I always like when they ask that question. The average day of research is really interesting because so I go into the lab, every morning, and then I have a specific set of experiments that I have set up. So it's not like I have experiments every single day, but I try to keep myself busy. We are a very fast-paced lab. So normally a day for me would look like I get into the lab, check my emails, because as a principal investigator, there is a little bit more of administrative tasks that I need to fulfill and make sure that my staff and the team is well, that there aren't any outlying challenges or conversations that need to happen. And then I go into the lab and usually I'm checking my cells. So most of the work that I do is going to be at the cell level. So I'm doing a lot of microscope checking. And then there is another part of the lab that does more of the animal model system. But in order to create those animal models, these cells need to be grown and a lot of the basic experiments need to happen in cells. So that's what I do. And I just do all the testing. So right now, some of the work that I'll be taking on in the next couple of weeks will be defining better parameters of our gene editing pipeline for the head and neck and esophageal. So my day will look like bringing up these cells from our head and neck cancers and then exposing them to these lipid nanoparticles that carry our CRISPR-Cas9, putting them in the cells, collecting the cells at 72 hours, and then doing a lot of molecular analysis, which would be PCR, gel electrophoresis, and sequencing. And that's pretty much what the day-to-day life of someone in our lab looks like. Those are pretty much most of the basic techniques that we're all doing at a given time. Sequencing is very heavy in our lab because it's the most reliant outcome to really gauge if our gene editing reaction has worked efficiently or not by sequencing.
Okay, I'm trying to find good questions over here. There are a lot in the Q&A.
Sure.
A lot. Let's see. Does CRISPR editing work at any section of the DNA, or do geneticists need to find specific nucleotide patterns?
No, so one of the things that is nice about CRISPR is that, well, yeah, we can find any site within the gene that we're interested in. But with Cas9, so depending on because there's many different types of Cas molecules or genetic scissors, and we just need to find a 3 nucleotide pattern, which is what we define as NGG, which is what the PAM recognizes, and that N can be any of the 4 nucleotides. So as long as the first is any of the 4 and then followed by a GG, the SP-Cas9, which is the one that we're utilizing, pyogenes Cas9, will cut the DNA along with any other, the 20 base pairs of specific site that you would like. So there is a lot of room for us to be able to cut the DNA at defined places. There's other gene editing technologies needed bigger and broader, bigger and more defined recognition sites, but with Cas9, it's just those 3 nucleotides. Therefore, there's many multiple options for it, and we test different sites within an area of interest before we define a specific guide RNA or specific cut site that we will stick to. Only from the context of discriminating between a diseased gene versus a non-diseased gene is where there is a more defined aspect of it, because maybe that NGG is not identified in the area where it's most optimal to target your gene. But in most cases, we can overcome that by other aspects of combinatorial approaches that don't really need to be limited by the gene editing.
That is fantastic. And I think given that we've only got, 2-3 minutes left, we might call it there. There are a lot of questions in the chat and a lot of questions in the Q&A that we didn't get to. We want to make sure everybody is aware that this recording, as well as Dr. Rivera Torres' slides, will be available at DelawareMiniMed.org tomorrow, perhaps the next day. So you will be able to view it again. You will be able to take notes. You can look at her slides. Tim, what am I missing? Anything? Tim, you're on mute, buddy.
I said, Dr. Torres, thank you very much. I think that's it, Kate.
Okay.
All right, so we will let everybody go. Thank you so much for coming to this first session, and we will see you next week.
Yeah, thank you so much. Have a great
Thank you,
night,
Dr.
everyone.
Rivera Torres.